to have you on Planetterrian Daily, episode 134, coming to you on July 28, 2026. Here's what's new in science, health, and longevity research. Simulations show water's local asymmetry drives proton hopping through clusters of 6 molecules rather than free drift. An international team led by Heidelberg University ran the most complex quantum simulations to date. Protons move by hopping across clusters of 6 water molecules rather than drifting as isolated particles. The modeling incorporated sites in Cambridge, Bochum, Dijon, and Heidelberg. The asymmetry creates directional preferences that standard diffusion models miss. The framework explains transport efficiency in aqueous environments at atomic resolution. Multiple collaborating institutions contributed computational resources to reach this scale of simulation. Now, here's something most people get wrong about how protons move through water. Right now, inside every cell and extracellular fluid in your body, protons hop from one water molecule to the next across small clusters. The latest quantum simulations show these hops occur within groups of 6 water molecules whose geometry creates a preferred path rather than random motion. The process completes in timescales far shorter than typical molecular diffusion, allowing rapid pH buffering across membranes. Any model of acid-base balance, enzyme kinetics, or mitochondrial proton gradients must account for this hopping mechanism rather than treating protons as conventional solutes. From the atomic details of water itself, we turn to a new molecular sensor that exploits water-based chemistry for imaging. Researchers developed HyBrick and HyBrick-2 by combining luciferase activation with bioluminescence resonance energy transfer. The sensors were tested in HK293T and HeLa cells with PiCTZ luciferin. HyBrick-2 improved brightness and responsiveness over the first version. The design enables excitation-free imaging with reduced tissue absorption in the red window. The approach offers a modular route to higher contrast bioluminescent calcium reporters. The same architecture supports future tuning of response speed while retaining the core energy transfer step. While HyBrick-2 watches calcium inside cells, another study asks how the first cell-like compartments could have formed in the first place. Hebrew University researchers showed that mixtures of fatty acids and hydroxy acids outperform either alone. The resulting assemblies resisted disruption better under early Earth conditions. Dr. Moran Frankel-Pinter and Dr. Rodem Edry led the experiments. The work addresses how simple prebiotic chemistry could produce cell-like compartments. The findings supply a cooperative mechanism for membrane emergence without modern lipids. Assemblies formed from both molecule classes maintained integrity across repeated cycles of hydration and dehydration. From how membranes might have first appeared, we look at how modern microbes can be guided to produce better membrane-targeting compounds. The team applied evolutionary-scale Cambrian embeddings to pardaxin and produced single and multi-mutant analogs. Principal component analysis combined with conventional design rules yielded candidates active against either E. coli or S. aureus. Molecular dynamic simulations linked alpha helical stability to selective intermembrane activity. Experimental validation confirmed the predicted specificity patterns. The work demonstrates how evolutionary machine learning can expand the pool of narrow-spectrum agents. The resulting variants retained activity while reducing off-target effects on non-intended bacterial strains. From engineered peptides that disrupt bacterial membranes, we move to natural membranes under stress in coral reefs. Woods Hole Oceanographic Institution researchers applied a medical-style diagnostic approach to coral systems. The surrounding water contains detectable markers before visible damage appears. The method parallels blood or urine tests used in human medicine. Early detection could allow intervention before bleaching or collapse becomes severe. The work demonstrates minimally invasive monitoring of reef health trajectories. Chemical and microbial signatures in seawater were cross-validated against conventional visual surveys at multiple reef sites. From monitoring external environments, we examine internal survival strategies that persist only under fluctuating conditions. Earth Life Science Institute researchers combined population modeling with comparative experiments. The unusual behavior persists over evolutionary timescales only when environments fluctuate. Constant conditions allow the strategy to be outcompeted. The Journal of the Royal Society Interface paper explains long-term maintenance of an apparently costly trait. The mechanism relies on temporal variation rather than spatial structure. Alternating nutrient-rich and starvation phases were required to maintain the trait across hundreds of generations in the model. From microbial trade-offs under varying conditions, we turn to whole animal energy trade-offs in seabirds. Nagoya University-led tracking showed increased reproductive output the following season. The same birds paid a survival cost. Researchers experimentally raised flight costs during breeding and followed individuals through migration and the next cycle. The trade-off appears only after high-demand seasons. The pattern links current year effort directly to future performance metrics. Individuals that expended more energy during breeding later migrated farther distances than controls. From animal thermal and energetic limits, we consider species already living near those limits. Lingnan University and international collaborators tested tolerance in species exposed to direct sunlight. Warming further compresses the safety margin between current maxima and lethal thresholds. The experiments quantified how little additional temperature rise the animals can accommodate. Results highlight vulnerability in ecosystems already at the edge of habitable ranges. The study focused on species that cannot behaviorally escape peak heat. Tolerance assays were performed on individuals collected from multiple intertidal zones during peak summer conditions. From species already at environmental edges, we look at how human movement of species creates new edges. Tel Aviv University researchers built the tool from traits of the more than 120 species already recorded since the Suez Canal opened. The model ranks likelihood based on biological and ecological characteristics. It identifies candidates for future invasion without waiting for first sightings. The approach offers an early warning system for managers. More than 150 years of canal-mediated movement provide the training data. Species with broader thermal tolerances and planktonic larval stages received the highest invasion risk scores. From modern species movements, we step back to ancient human movements and diets. Teams from the universities of Cordoba and Pisa examined human remains from a necropolis. Dietary traces showed clear differences by gender and social status. Mobility patterns emerged from isotopic signatures in the bones. Funerary practices reflected status through food-related symbolism. The study reconstructs daily life patterns that parallel recurring social structures across time. Isotope ratios from bone collagen and tooth enamel were measured across more than 50 individuals spanning different age groups. From ancient health and social patterns, we examine a more recent historical health catastrophe. Groundbreaking modeling traced the epidemic to the first fleet arrival in Sydney. The outbreak swept through First Nations communities shortly after British ships landed. Researchers documented a devastating demographic impact that altered resistance to colonization and land management capacity. The effects remain measurable in descendant populations today. The study reconstructs transmission patterns from historical records and population estimates. Mortality estimates reached as high as 220,000 individuals within the first year of spread. From large-scale historical shocks, we consider how everyday technology access shapes development today. UCLA psychologists found ownership itself correlated with poorer scores on standardized measures. Bringing the device to school did not change the association. The study examined elementary-age students and controlled for other variables. Results suggest the presence of the device at home may affect reading habits or attention allocation. The work supports examining device access patterns beyond classroom policies. Associations persisted after accounting for household income, parental education, and school-level reading resources. From individual development, we examine how collective aid structures affect institutional integrity. An SKKU-led team with collaborators from Chicago and Gothenburg used micro-level data across multiple agencies. The study is the first to quantify how simultaneous operation of many aid organizations correlates with higher corruption. Fragmentation creates overlapping oversight gaps that local actors can exploit. The findings point to coordination mechanisms as a potential mitigation lever. Data covered specific regions rather than national aggregates. Micro-level transaction records from several dozen aid projects formed the primary dataset. From data governance in aid, we turn to data practices in another field facing volume and consistency challenges. A qualitative study of Catalan researchers found inconsistent practices across document types and formats. Excavations produce large volumes of mixed records that hold scientific value. Current methods leave much of this heritage poorly organized or at risk. The Journal of Documentation paper calls for standardized approaches suited to digital workflows. The findings reflect broader challenges as archaeology shifts toward larger digital data sets. Interviews with field staff revealed variation in how photographs, field notes, and stratigraphic logs are archived. From how aspirations form in young people, we close with shifting occupational expectations. Researchers at Penn State tracked shifts in occupational expectations from 2000 onward. Family socioeconomic background influenced choices differently across job types. American students aiming for the highest status careers showed relatively small socioeconomic gaps. The patterns held across both countries despite differing education systems. The work highlights how aspirations have decoupled from some traditional background constraints. Longitudinal surveys captured responses from thousands of adolescents in repeated waves over 2 decades. Before we go, keep an eye on further tests of outbreak 2 in intact neural circuits and how the calcium sensor performs against existing red fluorescent probes. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. And before you go, this show is part of the NARA Network, a family of daily podcasts on tech, science, and markets, each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. OmniView, a balanced daily world news briefing that takes every side seriously. Find every show free at naranetwork.com. Following the long arcs, story trackers on naranetwork.com follow major programs across episodes. Tesla, SpaceX, AI, space science, and more. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.